So we've looked at mitosis, and meiosis, and just the first part of meiosis, really, and some of the terminology and the basics of it. We're going to be coming back to that as well. But what we're going to do now is kind of be going back and forth a little bit to kind of see the overlap and the connection, so it kind of makes, I think, a little more sense, instead of looking at them as completely separate things, to put them together. And we'll look at some of the inheritance patterns uncovered by Gregor Mendel. And so I'm not going to give the exact examples historically that Mendel used, partly just for aesthetic reasons, and that yellow and green, I'm using color here, and they don't look any different on this board. So I'll be using some different colors just as examples, but with the same principles. So let's talk about a couple of experiments. And some of these are hypothetical because Mendel didn't do it exactly like this, because he was working with pea plants, and then there are different colors for flowers, and there are different heights. They're sort of tall and short, and there are different colors of the pea pods, and then there are different textures to the peas themselves, smooth and wrinkled, and all these different things, and he assigned them as certain characteristics, and then the alleles would be the two versions of them. So like the wrinkled version versus the smooth, or the tall versus the short, or the green versus the yellow, and so on. Here I'm going to use flower color, and we're going to have this color over here represent red, and this color here represent white, even though it looks kind of blue probably to you. So what Mendel found is that if you took two red flowers and then cross-pollinated them, that he could get, all red flowers, and then those could be crossed with other red flowers and get all red flowers, and so on. And here, cross those with other red flowers, and then their offspring are all red, and so forth. And so, he was finding this as one particular pattern, say 100%, 100%. With white flowers, same sort of thing. So you get the white flowers, cross them with other white flowers, 100% of those, all white, cross these offspring with other white flowers, plants, obviously with white flowers, and then get another 100%, white, and so on. So these groups of plants that had a trait that could be then crossed with others with the same trait and produce all offspring with the same trait, and then those offspring would have offspring with the same trait, and so forth, that would be called true breeding. And to apply some more of the modern terms to it, true breeding plants are going to be ones that are homozygous. And what that means is that when we have the different versions of alleles, the homozygous ones, both alleles are the same. Okay, so both alleles are the same. Now, Gregor Mendel didn't know about this, didn't know about the alleles and all, but that's the idea of what's going on. So, what then became more curious, okay, is, and that's just the idea, the first part of it. So just kind of look at plants, different characteristics, breed ones of one characteristic, and then you end up finding that they can have offspring that are the same as the parents. So there's nothing exciting or to really question about that. It's just that traits are inherited by the offspring from their parents. What was more curious? Okay, it's this. In doing some of these breeding experiments, Mendel then found something like this. He would take two red flowered individuals, let's say, and he would find that some of the offspring were red and some were white. And these are white here. And now that would be curious. So this would be the parent generation, so the P generation, and this generation would be called the F1 generation. And the F1 generation, we find this. And so And the numbers, so he would count them. So that was one thing that Mendel did set him apart, is that he was also a mathematician, and he counted and kept very good records of the data that he was collecting and used it to do all the calculations that we talk about. That it was roughly 75% of them were red and 25% white, which was very curious. Now, if the white ones were bred with other white ones, they would lead to 100% white. But if the red ones, so this is times other white ones, if the red ones in this generation were bred with other red ones, you would get a combination of, depending on potentially the individual that was chosen, you get the same sort of thing again, you get 75%, or other sort of proportions, which we'll get into later. And that was curious. So what Mendel started to think is that there could be some characteristics or traits that could be hidden and others that would then be expressed. So that was the idea for having the dominant versus the recessive, so that some traits would be hidden, that the traits would separate like particles passed on to the offspring, and that there was a chance that you would get one type of particle or the other, and that some individuals, the true breeding ones, both of these particles, and this is the case we're talking about the alleles, for a certain gene, would be the same, and in other cases individuals could have two different types. So what we then need to do is start to assign some kind of notation to it. So the red, who are dominant, we're going to give a capital R, and the white, who are recessive, will have the lowercase r. So now what we have, let's say a parent who is true breeding and red. So this is the genotype, meaning we're looking at both alleles here, and they're the same. And then the phenotype is what it looks like, and that is red. And let's say we cross that individual with a plant that has white flowers. So the white now in this case is the phenotype. The genotype also has to be the true breeding, so they both have to be the same. And in this case, the white is being seen as the recessive trait because it can be hidden and then just appear later on. Mendel is known for then putting together the layout to look at the assortment of the genes and segregation of these alleles with a Punnett square. So what that means is that if this allele can be given to the offspring within one of the gametes, so we have to go back to thinking about meiosis and how the gametes are only going to have one of the two because they're going to be haploid. So remember the original parent is diploid, but the gametes are haploid. So this parent could give this R, capital, the dominant for red, or it could give the other 50-50. So 50-50 chance it's going to give one or the other, but the other one's exactly the same. This parent over here, it has two alleles. But again, it's a true breeding organism. So all of its offspring are the same, and its offspring's offspring are the same, and so on. So that means both alleles are going to be the same. We're designating it as the recessive allele because it's the trait that can then be hidden or masked. And so it can give either the lowercase r, the recessive for white, or really the recessive for white. That's pretty much the only option it has. And so this will make up our square. So that means this is parent 1 here, this is parent 1 here, this is parent 2 here, and when they mate, this gamete combines with this gamete, or this one with this one, or this one with this one, or this one with this one. And it really doesn't matter. In this particular case, you're going to have all the same outcomes, and 100% of them are going to be red. And that is what Mendel saw in this first cross. Okay, that all of them were red. And then he saw that he could take, two red individuals from this group, mate them with one another, and then find this ratio, the 75% red, 25% white, or essentially a 3-to-1 ratio. And that's going to be explained by, again, looking at the genotypes. So this genotype is heterozygous. So it's going to have the dominant allele as 1, and then it's going to also have the recessive allele. So even though it has the recessive allele, it's going to look red. That's because it's dominant. Any other individual that it's crossed with in that population is going to have the exact same genotype. Like this. And so as we then cross these individuals, we have several possibilities for the outcome. Right? You can put the dominant reds together and it's still going to look red. You can put the dominant with a recessive and it's still going to look red. Here you can put a dominant with a recessive and it's still going to look red. But here a recessive and a recessive can actually come together and have it look white. And see, that would be 1, 2, 3 to 1. And the data from his work with plants then supports this pattern, and it did so with all the different sorts of traits. So when he started to use flower color, or the color of the pea pods, or the texture of the pea pods, or the heights of the plants, all those different traits ended up having the same basic patterns of inheritance. And so because of this, we then have Mendel's law of segregation. And so for the law of segregation, what we have is that there are two versions of a trait. And they separate into gametes independently. So if a parent if a parent has, the same, two of the same or two different, there's no difference in the chance of one being passed on to an offspring more so than the other. Meaning essentially the dominant one doesn't have a better chance of being passed on than the recessive one. They have an equal chance, of being passed on, and which one is passed on is completely random. So it could be the dominant or it could be the recessive, 50-50 chance. And it's always going to be the case. Right? And that's essentially the idea behind the law of segregation. And how that ties into our meiosis is if you go back and look at the pattern of the sister chromatids separating, if you had two different versions within the original cell of one allele being different than the other, one being dominant, one being recessive, then you would have an equal chance of them being passed on within the gametes. You'd have equal numbers of them in the gametes. And that's sort of the idea of this first part. What we then need to start to look at is, this is only just one trait or one characteristic. What about multiple characteristics? When we have several different characteristics Can they be tied together? Or do they act independently? How does that all work? And so that'll be the second law that we'll get into next, which will be independent assortment. And with that, we're also going to tie in another feature of meiosis, which is one of the real characteristic features of meiosis that, again, increases genetic diversity, and that's called crossing over. So that will be the next thing that we do.